Hi, my name is Brody Quinn.
I'm Alexis Hand.
I'm Melissa King.
And I am Holden Wood.
And this is our presentation on the comparison of microbiome diversity of Canis lectrans, also known as Coyotes, fecal matter in Monterey County. Coyote populations have been under threat by issues from urbanization and the threat of their normal diet becoming distorted with easier access to anthropogenic foods. Anthropogenic foods are human polluting foods found in trash that are causing severe microbiome influences in large metropolitan cities. This is discussed more in Scott Sugden's scientific journal titled "An Altered Microbiome in Urban Coyotes Mediates Relationships Between Anthropogenic Diet and Poor Health." The data was collected from 2 different locations in Alberta, Canada, that contained urban versus rural housing of the city to analyze the changes in coyotes from both locations. After they were dissected and samples were collected, the urban areas saw more severe cases of malnourished coyotes that had indicators within their bacterial composition. Additionally, in a research article by Michelle J. Russell researched the impacts of coyote species on the agricultural industry. They noticed that although the strains of Shiga toxin producing E. coli they were looking for was not present, they did discover other pathogenic E. coli and Salmonella strains that were present that were produced from the improper practices of mitigating microbial contaminants on the crops. According to our hypothesis, we predict that the Canis lectrans fecal matter will provide evidence that species found in Monterey County are going to have healthier digestive tracts compared to those that are discovered scavenging around suburban and large human populated environments.
To start our research, feces samples from coyotes were collected on January 22nd, 2020 in the trails on Engineer Canyon Road in Marina, California. Samples were added to a 3 milliliter LB broth and were incubated at 37 degrees Celsius for 24 hours. Unknown samples of coyote scat were available and categorized by appearance based on if there were bones, fur, twigs, or moss present. Using aseptic techniques, the unknown bacteria were isolated from the broth and streaked onto LB plates. The plates were then incubated at 37 degrees Celsius for another 24 hours. Aseptic techniques allow for a contamination-free environment for the bacteria to be isolated. Next, bacterial DNA was extracted by mixing a CFU, or a colony-forming unit, with 20 microliter solution into a tube and placed into the thermal cycler for 15 minutes. PureTac PCR bead tubes were prepared with 23 microliters of master mix, which was made of 1 microliter of 27F primer, 1 microliter of 1492R primer, and 2 microliters of water, and 2 microliters of microlysis in a CFU, and once again placed into the thermal cycler. Standard procedures were followed to prepare the 10% agarose gel. Then 5 microliters of sample were mixed with 3 microliters of dye, loaded and ran at 120 volts for about 35 minutes. DNA was isolated by using the PCR reaction cleanup method using the PureLink PCR purification kit. The DNA was sent and sequenced at the Molecular Cloning Laboratory in San Francisco. The sequences were then inserted into the NCBI database using the BLAST, specifically the nucleotide BLAST, to compare and identify the bacterial DNA. Then nitrate reduction test was executed by inoculating a broth that had a Durham tube and incubated at 27 degrees Celsius for 48 hours. If bubbles were present, the nitrate was reduced to nitrogen gas. If not, then drops of reagent A and B were added. If no change after 10 minutes, then a pinch of zinc powder was added. If the broth turned red after the addition of reagents A and B, then there is reduction to nitrite. If the broth turned red after the addition of zinc, then there was no reduction. To perform the anaerobic test, standard aseptic techniques for streaking and inoculating broths were performed. 1 nutrient agar plate and 1 inoculated LB broth were placed in the aerobic conditions to grow at 37 degrees Celsius for 24 hours. A different streak nutrient agar plate and inoculated LB broth were placed into the gas pack E2 anaerobic container system to grow under anaerobic conditions. The Kirby-Bauer was performed by comparing the broth to the McFarland standard, then diluting the broth containing bacteria with saline until an absorbance reading of 0.239 absorbance was obtained using the spectrometer set at 600 nanometers. 100 microliters of diluted broth was pipetted onto a plate and spread around with a hockey stick and turntable. 8 antibiotics, including streptomycin, amoxicillin, gentamicin, penicillin, chloramphenicol, tetracycline, neomycin, and erythromycin, were placed onto a plate and then incubated at 37 degrees Celsius for 48 hours. After incubation, the zone of inhibition, which accounts for the bacterial abilities of resistance to the antibiotic, were calculated and recorded. Then we took the data collected and input it into Excel to create the graphs for our project.
In this graphic, it shows the frequency of different genus of bacteria observed in the fecal samples. After viewing our sample data, the majority of bacteria seen in the sample was gram-negative Escherichia with a percentage of 47% abundance in fecal samples. Second was the gram-negative Acinetobacter with a percentage of 29% abundance. The final bacteria that was seen in the fecal samples that was worrisome was the gram-positive Bacillus bacteria. That was about 12% of the proportion. This figure shows the results of antibiotic resistance. We can see the different antibiotics and their zones of inhibition for bacteria in the fecal samples. This figure gives us insight on what foods coyotes are consuming and how it is seen through the fecal samples. Most foods coyotes are eating are creating a resistance to basic types of antibiotics. This occurs from the foods that have been beneficial of resistance, those of bacteria. In our graphic provided, we can see that all of the bacteria are resistant to antibiotic penicillin, showing a 0 millimeter zone of inhibition. The results of the nitrate reduction test revealed that 8 out of the 8 of the bacteria tested had no nitrate reduction abilities, as well as 8 of the bacteria were able to reduce nitrate to nitrite. And 2 out of the 8 of bacteria had the ability to reduce nitrate to nitrogen gas. The abilities of bacteria to perform denitrification can be an indicator to the overall diet and health of the coyotes. It has been found that the high amounts of nitrate and isotopes of nitrate reveal that the main source of nitrogen is coming from inorganic fertilizers. Due to the various surrounding agricultural fields, this could be a major source of nitrogen finding its way into the digestive system of the coyotes and Salinas. Therefore, it would be beneficial for the coyotes to carry bacteria that have nitrate reducing capabilities because most of the time, nitrogen and oxygen are present together in the form of nitrate. If the nitrate is not reduced down to nitrogen gas, then it remains present in the environment, which can pose major problems for the groundwater.
Okay, so as my group member Melissa mentioned shortly ago, the most abundant genus present in Monterey County coyotes' gut microbiota are gram-negative Escherichia, gram-negative Acinetobacter, and gram-positive Bacillus. All of these bacteria can thrive under low nutrient anaerobic conditions that are consistent with the bacteria we'd expect to see present in healthy coyotes from rural populations similar to Monterey County, which I believe would be regions of Canada and Texas for our study. As seen here in the image on your left, for healthy coyotes, there's increased fusobacteria, increased anaerobiosporellum, and increased Sufferella, which are all phyla possessing anaerobic capabilities, just like the most abundant bacteria present in the guts of our local coyotes. The image also shows an increased protein in their diets with a decrease in anthropogenic foods, being human scraps and leftovers, which is consistent with the Bacillus bacteria present in our sample, as Bacillus are typically found in decaying items like red meats and rotting roadkill, which indicates our coyotes are consuming less human scraps and protein diets on a regular basis. Although, of the bacteria present in our fecal sample, the only bacterium of concern is Bacillus bacteria because they're typically always pathogenic, and too much of this particular bacteria present in the gut biome can cause coyotes and other organisms severe health issues. The fecal sample, when analyzed by Kirby-Bauer, indicates a 0 zone of inhibition for the antibiotic penicillin. This is generally what we'd expect as two of the most abundant genera present in our coyote's gut biome were gram-negative, and penicillin works primarily against the thick peptidoglycan layer present in gram-positive bacteria. My group and I then thought, were our bacillus bacteria present resistant to penicillin too? But upon reviewing the results, there were outliers present showing zones of inhibition for the antibiotic, which we'd say bacillus are likely the outliers, showing resistance to penicillin. Lastly, to provide a bit more evidence, Monterey County coyotes are healthy given their wide ranges of habitat. High amounts of nitrate and nitrate isotopes are found to be sourced from inorganic fertilizers common to our local agricultural fields. So we believe it's easy for these forms of nitrogen to find their way into the coyote's digestive tracts. So it's beneficial for our coyotes to have nitrate reducing abilities, which the majority, 55% of the bacteria do, supporting our claim for healthy rural coyote populations in the Monterey County. And I believe Brody will take from here. Thank you.
Thank you, Holden. We originally predicted that the Canis latrans fecal matter will provide evidence that species found in Monterey County are going to have healthier digestive tracts compared to those that are discovered scavenging around suburban and large human populated environments. The Canis latrans fecal matter provided sufficient evidence that Monterey County species have healthy digestive tracts compared to those that are discovered around urban environments. As discussed by Melissa and Holden, the most significant genus of bacteria was Bacillus, which is acquired through the consumption of nitrogen-rich organisms, such as dead animals or untreated plants that contain infected soil. For future research, samples would need to be taken from different parts of the coyote's digestive tract for accurate analysis on where the microbiome is playing a large role in keeping the body healthy. Many sources that looked into the evolution of coyote populations due to urbanization emphasized the importance of observing changes in the small intestine rather than the byproducts of the food broken down after it is excreted. Furthermore, there haven't been any sources based on the Monterey County that provide a basis to compare our data over time. So being able to keep track of this data on a 2 to 3 year collection can provide a better graph for change over time. Lastly, using expensive tracking equipment could keep a track of the frequency of coyotes venturing into suburban areas around the coast of Monterey and see why they prefer to remain in these areas rather than returning to the more protected regions like Big Sur State Park. Here are references for our work. And lastly, we'd like to thank Jennifer Cato, Melanie Yu, Arlene Hoffa, Caitlin Tatsukawa, Rachel Durand, and Lou Zyberg for their efforts in providing our research group with the help we needed to complete our work. Thank you for all of your help.